Welcome to this cell projects presentation about how to perform adherent cell electroporation using the InSitu Porator. This image shows what you will be able to do. The fluorescent dye, lucifer yellow, was introduced into cells growing on a transparent and conductive glass up to this line. The line is easier to see under phase contrast. Here it is. You can see that the dye has diffused through gap junctions from the cells on the left, which have been loaded with dye by electroporation, to the cells on the right, resulting in this gradient of fluorescence. The InSitu Porator consists of three main components: the pulse generator, the chamber holder, and InSitu chambers which come individually wrapped in sterile packages. The chamber is made up of three parts. A glass slide that has a conductive coating on it forms the base. Bonded to this is a plastic part that has interior dividers. There's a clear plastic lid on top for sterility. The manual has a complete set of diagrams that show the chamber in detail and describe how the conductive coating has been etched to provide adjacent regions of electroporated and non-electroporated cells. Here's what you need: PBS, formaldehyde, DMEM without calcium with dialyzed serum, DMEM without calcium, waste beaker, lucifer yellow, pipetters and pipette tips, chamber holder, InSitu Porator, as well as some tissues for handling and cleanup. Cells are trypsinized, then transferred into a tube and centrifuged. It is very important for the cells to be well spread as they grow in the chamber. For this reason, it is best to centrifuge the cells to remove any traces of trypsin. The medium with traces of trypsin is suctioned off. Cells are resuspended in approximately 1 milliliter of growth medium and then plated in the chamber. The pipette contains the cell suspension. Remove the lid from the chamber and pipette the cells dropwise into the chamber. To make it easier to carry around, the chamber is placed in a 6 centimeter petri dish. The chamber goes back in the incubator for the cells to grow. The plated cells will spread well and they will grow to touch each other to form gap junctions. When the cells are ready to electroporate, set the InSitu pulse generator to the desired conditions. Refer to the manual for a description of the simple setup process. When the voltage on the InSitu Porator is set, the red light on the keypad will turn on. Take the two warmed bottles of DMEM without calcium out of the water bath. One is DMEM without calcium, but with dialyzed serum, and one is DMEM without calcium. Have the pipettes ready. Load lucifer yellow into the pipetter and set it aside. Take the cells out of the incubator. Remove the medium by inverting it in the wash beaker at first, then dabbing it onto the tissue. Wash once with DMEM without calcium. Do not try to remove absolutely all of the DMEM because the cells may dry. Add the lucifer yellow carefully onto the barrier so as not to disturb the cells, but quickly enough so that the cells do not dry. Use 200 microliters for each half of the chamber. Put the lid on the InSitu chamber, then load it into the slider. Make sure that the end tabs of the chamber fit into the slots of the slider. By pushing the slider, insert the chamber into the chamber holder. Push the pulse button on the pulse generator keypad. The LED on the pulse generator and on the chamber holder will flash red during the electroporation process, and then flash green to indicate the InSitu Porator cycle is complete. Pull the slider back out of the chamber holder and remove the chamber. Aspirate most of the lucifer yellow to use it again, but take care not to dry the cells. Add DMEM without calcium with dialyzed serum. The serum helps the pores to close. The chamber goes back into the incubator for the transfer of lucifer yellow through gap junctions to happen. Timing must be consistent from experiment to experiment. The standard is 3 minutes. Pour off the medium into the waste beaker. Add 4% formaldehyde fixing solution with a Pasteur pipette. Wait at room temperature for at least 2 minutes. Pour off the formaldehyde and rinse a couple of times with PBS. The cells are fixed to the glass, so there is little danger of them detaching during washing. To be able to see the cells under phase contrast, it is important to place a cover slip on top of the chamber. Fill the chamber with PBS, forming a proud meniscus. Avoiding air bubbles, place the cover slip. Don't worry if the PBS overflows, just absorb the extra PBS onto tissue. You need an inverted fluorescent microscope, preferably with phase contrast and with a filter block for lucifer yellow. Under phased contrast, look for the edge of the electroporated area. You will see 2 lines. Under fluorescence, cells on half of the slide will be fluorescing intensely. The line closest to the glowing cells is the edge of the electroporated area. Any fluorescence beyond that is due to the transfer of lucifer yellow through gap junctions. For example, this image of epithelial cells shows cells that have been electroporated on the left side of the image, transferring lucifer yellow to neighboring non-electroporated cells, creating a gradient of glowing cells from the edge of the conductive coating toward the right. This image shows the same frame of cells under phase contrast. Note that you can see the etched line very clearly. A quantitative measure of how much gap junctional transfer has occurred can be done manually by working with the fluorescent and phase contrast images of electroporated cells. The stars along the edge of the conductive area are considered donor cells. They have been electroporated and taken up lucifer yellow. They pass lucifer yellow to neighboring cells to the right, which pass it on still further in diminishing amounts. The ratio of the dots to stars, 41 to 8 in this case, can produce a number for comparing how extensive gap junctional transfer is. In this case, 5.1 is the gap junction index. An alternative to manually marking and counting cells is to use image analysis software to measure the relative amounts of glowing along the non-electroporated side of an electroporated cell border. These cells do not have gap junctions. Here is the right edge of the electroporated area. Note there is no gradient of fluorescence, which indicates the absence of gap junctions. Under phase contrast, you can clearly see all the cells. Note that the etched line is also clearly visible. In this example, all of the cells in the chamber were stimulated with epidermal growth factor, EGF, and 10 minutes later fixed and probed for phospho-ERK. Note that the stained cells on the right have not been electroporated, and the cells on the left, up to this line, have been electroporated. The non-electroporated cells show brown staining with a phospho-ERK antibody and immunoperoxidase. The electroporated cells show no staining as the electroporated phospholipid has blocked the SH2 domain of GRB2 and inhibited ERK phosphorylation of EGF. Note the region of 3 to 4 cell lengths to the right of the line, where cells were not electroporated yet do not show staining. These are cells that have acquired blocking peptide from their electroporated neighbors via gap junctions. This is the most powerful demonstration possible that the inhibition of the signal must be due to the peptide and cannot be an artifact of electroporation, since these cells are not electroporated. This is the same frame shown under phase contrast. Note the absence of any change in morphology after electroporation. For further details regarding how to obtain optimal electroporation, please refer to the hard copy manual or the InSitu electroporation section of the Cell Project's website. You will find a number of helpful resources there, and you are always welcome to contact Cell Projects with any questions.